Hi, welcome back. We are now moving on to the most abundant of those formed elements that we saw in the breakdown of whole blood. We're going to talk about red blood cells. So remember, they are 99.9% of the formed elements, of that 45%, which is of the whole blood. So when we take a look at red blood cells, their main job is to transport oxygen. That's it. There's some lesser roles of transporting carbon dioxide, but really, the job of your red blood cells is to bring oxygen from your lungs to out the rest of your tissues. That is its primary role, and its structure and internal filling, if you will, is adapted greatly to be able to do that job very efficiently. So if we're going to take a look at a single drop of blood, you can kind of see my vampire blood splatter there. A single drop of blood has about 260 million red blood cells. Now, I know drops vary in size, but just like an average, drop of blood, 260 million red blood cells. That's crazy. There's a measurement that we call the hematocrit. So the hematocrit is the percentage of your whole blood that is red cells, and sometimes it's called packed cell volume, PCV, which is the same, almost just the same as our formed elements, because remember, 99.9% of our formed elements are red blood cells. So when we say, that percentage is 45%, a hematocrit value is not going to be much more off than that. So 45% plus or minus a few percentage points would be an acceptable hematocrit, the percentage of whole blood that is red blood cells. So take a look at what red blood cells look like, kind of super duper close up. You probably are familiar with these, may have seen them before. So this is erythrocyte is their technical term. You can either use erythrocyte or red blood cell, that's fine. Erythro means red. Oops, get a pen going. This means red and this means cell, so it basically means red cell, so red blood cells. So they're what's called a biconcave shape. So concave, remember, dips in. Bi means it dips in on both sides, like a little donut that hasn't been punched through all the way in the middle. So this shape, so remember I talked about how they're very well adapted to this central role of carrying oxygen. This shape does a couple things. So one, it allows them to move through the capillaries easier. They're bendy, right? They're not very rigid. And so capillaries are really small. The blood vessels are only about has a diameter just a tiny bit bigger than the red blood cells themselves. So we can kind of see how they can have to squeeze and twist through these small blood vessels. Another benefit of having this biconcave disc is it allows them to have more surface area because that oxygen has to diffuse into and out of the red blood cell. So if we just had a shape like this, we would have only so much surface area, but we put a little, a divot in there, and we're going to increase the surface area that we have for that diffusion of oxygen from the lungs and release of oxygen at the tissues. And then lastly, allows them to stack. So we have the flexibility, we have the surface area to volume ratio, and then as you can see in this picture, they can stack and almost move like a train through those capillaries. So they're flexible, they're stackable, and they have a high surface area to volume ratio. All of those things contribute to the efficiency of the red blood cells carrying oxygen. Okay. Also, a very key feature in erythrocytes is they lack a nucleus. They are the only mature adult cell that lacks a nucleus. So remember, eukaryotic cells, we are organisms made up of eukaryotic cells. The definition of a eukaryotic cell means that it has a nucleus. Now, this doesn't mean a red blood cell is a prokaryotic cell. No, it starts off with a nucleus. So we're going to see that in the development of red blood cells in a little bit. It just ejects its nucleus when it becomes mature. Because again, the only thing the red blood cell really needs to focus on is carrying oxygen. If you have a big nucleus in there holding DNA that doesn't serve its purpose, then that's just kind of a waste of space. So the cell produces as much hemoglobin as it possibly can. Once its cell is packed full of hemoglobin, the nucleus hits the road, and then the rest of the cell is just this big sack of hemoglobin that serves its purpose really well of carrying oxygen. But because of that, you've lost your ability to repair anything because you've just kicked out all of your genes. You can't make proteins to repair any damage. So once a red blood cell is mature, it only can last circulating in the body for about 120 days, which is like 4 months. So it circulates until it kind of gets worn out, and then our body recycles it. And we'll be also talking about that in a couple slides. So let's take a look at this hemoglobin. What is this protein that is jam-packed inside of the cell? It is a quaternary structure protein because it is made up of 4 tertiarily folded, right? Primary, secondary, tertiary, quaternary, right? So each one of these subunits is a folded amino acid chain, which is our protein chain, if you remember your protein synthesis from general bio, and then 4 of these protein chains hooked together to make a complete hemoglobin molecule. And we have 2 alphas and 2 betas. So alpha chain 1, alpha chain 2, beta chain 1, beta chain 2. So we basically have 4 big blobs of hemoglobin. Inside each one of these, in the middle of each one of these, it's kind of like this little disky thing here. This is what we call the heme. So the heme of hemoglobin, so there's that one hiding back there, and then the other one's here. So heme is this disky looking thing. It's the iron containing core. And then the globin is the protein chain, right? So all of the chains of amino acids, because it's kind of globular, like there's a protein structure to it. So if we take a look at what the heme is, so the heme is this big structure. It's a very large organic molecule, but look at what's right in the middle. So you know what Fe is, what the chemical symbol for? This is iron, right? So we know blood is iron rich. This is where the iron is. So that's kind of the little red circle in that disc is representing the iron core. And then we have just a pretty large organic molecule. You'll notice there's carbons, nitrogens, some oxygens and hydrogens in there, but the iron is the core, and it's positively charged because oxygen tends to have sometimes a negative charge to it if it's ionized. And so oxygen and iron are really attracted to each other. So if we take a look at a full-on hemoglobin molecule, a complete hemoglobin molecule, we have 4 subunits, 4 heme molecules, 1 oxygen molecule combined to each heme. So how many oxygen molecules can we carry per hemoglobin? 4. And this is what we call oxyhemoglobin right here. Oxyhemoglobin means we have oxygen. Oh, that's not a good color. Maybe yellow. Oxygen here, oxygen here, oxygen here, oxygen here. Those little red dots are the heme. So in a fully saturated hemoglobin molecule, you have 4 oxygen molecules per hemoglobin. And when oxygen is unbound, we call it deoxyhemoglobin. Here's that kind of abbreviation there. And what I want you to do is take a look at the shapes. I'll erase my little squiggles of oxygen here. But look at the difference between the oxyhemoglobin and the deoxyhemoglobin. And if you just kind of stand back a little bit, you'll see that there's a little bit of a difference in the shape between when oxygen is bound to the hemoglobin and when it is not. And that's a very important characteristic of the hemoglobin molecule. The nature of oxygen binding to a heme slightly changes the shape of that chain, the globulin chain. And by changing the shape of that globulin chain, it changes the affinity of the other 3 globulin chains. So what that means is, let's say you are at a hemoglobin at the lungs, right? So you don't have any oxygen on you, bound to any of your hemes. You show up at the lungs, high concentration of oxygen at the lungs, and then you get an O2 molecule binding right here to this first one. Okay? What that'll do is it'll slightly kind of change that first chain. And then what that does is it makes the next chain slightly more, or it makes all the other chains slightly more attractive, or what we use the word affinity. It's got a stronger affinity for oxygen. So then let's say the next oxygen gets bound up really fast, and then it's going to change its little shape a little bit, which means the other 2 remaining will have a stronger affinity. So then we get our 3rd oxygen, which is going to change its shape, and then our 4th oxygen, like ding, bang, bang. So the first one is kind of slow if we're going to do a rate here, and then we do a quick increase in binding. So if this might be our first oxygen, our second oxygen, our third and fourth. So it increases the affinity once you bind the hemoglobin, once the oxygen binds the hemoglobin. And then the reverse is also true. So let's say that hemoglobin now leaves the lungs, goes back to the heart, pumps out to the rest of the body, and now it's at the tissues where oxygen levels are low. So an oxygen will leave one of the hemoglobin subunits to go out to the tissues. And once we lose, right, so if we're going to go the other direction, it's going to be like this. So here's our first oxygen that we lose, so that falls off, and then the next one is going to fall off, and then it reverses that binding. So I'm not sure if I'm making sense. I feel like I'm stumbling over my words. So when that first oxygen pops off, it changes its shape back to being a deoxyhemoglobin, which triggers the other hemoglobins to be less bound to their oxygen, which means they're going to drop theirs off, which means the other last two are going to drop theirs off even quicker. The reason why that's important is because what do we want to do with the tissues? We don't want to forever hold on to the oxygen and never let go. That's not the point. We want to bind oxygen when we are at high concentrations, which is in the lungs, and we want to let go of our oxygen when we're in low concentrations, which is at the tissues, because that's the whole point of the red blood cells and hemoglobin is to pick it up at the lungs, drop it off at the tissues. So that whole conformational change of the protein itself really, leads into that functionality of hemoglobin. And again, we have 200, let's see. Oh, each red blood cell contains 280 million hemoglobin molecules, and we had 260 million red blood cells per drop of blood. Here on me. So we can do a little bit of math. You don't need to know this math, but it's just kind of an interesting formula. If we have 260, I'm running out of room, 260 million red blood cells times 280 million hemoglobin molecules per red blood cell, that gives us 7.28 times 10 to the 16 hemoglobin per drop of blood. What? So you know what 7.28 times 10 to the 16 is, right? So it's a 7.28 and then 14 zeros after that number. Hemoglobins per drop of blood. But how many oxygen molecules on each hemoglobin? 4. So we got to multiply that number by 4, which comes out to be 29.12 times 10 to the 17 oxygen molecules per drop of blood. Now, if you really wanted to find a big number, you can calculate what volume a drop of blood is. Is it like a milliliter, a half a mil, a tenth of a mil? Multiply that by 5 liters, and then you'll get how many oxygen molecules in a adult human blood volume in 5 liters, which is, I'm guessing, is going to be a really, really, really big number. So we've got a lot of oxygen circulating around our body thanks to the efficiency of the red blood cell shape and thanks to the efficiency of the hemoglobin molecule and its ability to bind oxygen very efficiently and drop it off when needed as well. It's not a permanent binding, it's a temporary binding of that oxygen. Okay, the last piece I wanted to talk about on this slide is this microscope, or on this, yeah, I guess it's a slide, it's a PowerPoint slide, looking at this microscope slide picture of these red blood cells. Now, if you'll notice, some of those red blood cells don't look biconcave shape. We've got these really weird elongated ones that don't really match. So this would be more of like a normal shape. These are more of the normal ones that I've drawn the arrows to. So what's going on with these weirdly shaped cells? Well, this is a slide of a blood sample from somebody with sickle cell anemia. So sickle cell anemia is a genetic problem where there's a mutation in the amino acid chain, those alpha and beta chains of the hemoglobin molecule, that when in low oxygen environments, like in the periphery or during exercise, or maybe sometimes just getting up and walking across the room, the hemoglobin molecules, all of the hemoglobin molecules are misshapen. And so then if you have 260 million hemoglobin molecules per red blood cell and all of them are misshapen, the whole red blood cell becomes misshapen. And so if you have blood going through your blood vessels that are supposed to go smoothly because of their very specific shape, they become misshapen, you're going to have a really hard time sending the blood cells through your blood vessels. And so things get kind of clogged up. It's inefficient. The tissues are starving for oxygen. So that's the anemia part of this disease, is because they don't do a good enough job carrying oxygen because of that misshapenness. And it is a recessive trait. So if you had two heterozygotes, right, so a little bit of genetics review, there's a 25% chance that your offspring would have sickle cell anemia, and a 75% chance that they wouldn't, but a 50% chance that they would be a carrier, which then could pass on that trait. Okay, a little bit of kind of by 211 genetics review there. All right, so let's take a look next at what happens when those 4-month-old red blood cells start to wear out. So this is showing hemoglobin recycling, which is, red blood cell recycling, but red blood cells are just big sacks of hemoglobin. So we're really just going to focus on hemoglobin recycling. So let's say you are a brand new baby red blood cell. You get born out of the bone marrow. You live your 120 days, right? So that's how long you can go before you start wearing out. About 10% of red blood cells will just hemolyse on their own. They just break down. They pop and they dump their contents into the bloodstream. Some macrophages will come along and gobble that up. Any hemoglobin that's released in this way just gets peed out, gets recycled, or not recycled, but it just gets peed out in the kidneys. The most of your red blood cells is 90% after their 120 days. Macrophages are recognizing that's a worn out red blood cell. Before it even gets to the hemolysis stage, it'll engulf it. It'll break it down, fuse with a lysosome, recycle the amino acids, so the globulin part, right? So the globulin part gets recycled back to the bone marrow. Okay, so that's the alpha and the beta chains of amino acids get recycled back to the bone marrow. And then what we're left with is the heme, right? So if you remember, the heme was in the middle, and it was kind of that organic molecule with the oxygens and the hydrogens and the nitrogens that kind of look like a flower. So the heme, or the iron, is removed, and that's this pathway right here. So the iron gets removed from the heme and goes back to the bone marrow. So far, we've done a pretty good job of recycling. We have recycled the amino acids from the globulin part, and we've recycled the iron back to the bone marrow. All of this organic part of the heme, we don't necessarily recycle that. We use it, it's more of a waste product, but it kind of does some things on its way to becoming a waste. So in the macrophage, this is converted into a greenish type of molecule called biliverdin, which is then converted into a yellowy type of molecule called bilirubin. And then the liver takes that bilirubin and uses it. So some of it as actually incorporated into a digestive secretion called bile, so that goes into the digestive system here. Some of that bilirubin just gets dumped back into the bloodstream and is filtered out at the kidney here. Some of these bilirubin that goes into the digestive tract from the bile gets reabsorbed into the bloodstream, but then gets excreted out of the kidney. And then further down in the digestive system, those turn into what are called urobilins and stercobilins. And so it's kind of interesting because the release of the bilirubin and the bilirubin-derived products from the digestive tract is what makes pee yellow. Okay. I don't have a brown. But these urobilins and stercobilins are what make feces brown. Okay, so it is a recycling of your blood, specifically the heme part of your hemoglobin, that gives you color to your urine and your feces. Now, another thing I want to point out with this whole recycling of red blood cells, and primarily the heme in the hemoglobin, is think of about the last time you got a bruise, like a really nasty bruise. So you whacked your shin bone on something, or you fell during skateboarding or ice skiing or whatever. So you get this big bruise, and it first appears as kind of a purpley, plumish, reddish color, right? And what is a bruise? A bruise is bleeding into the surrounding tissues, like your subcutaneous fat or your dermis. So you have blood leaking into those tissues. You get the repair, so you're not bleeding, you don't have internal bleeding anymore, but now you have this big pool of blood outside of your blood vessels just sitting in your connective tissues. Well, it's going to go through the same recycling. We have these macrophages are found in connective tissues, so they'll gobble up all of your red blood cells, break them down, try to recycle the best they can, but they will be converting that organic molecule to the biliverdin and the bilirubin. So think about the progression of your bruise. It starts off as being purpley reddish brown because that's the color of freshly bled blood. Then what color does it turn? It kind of starts turning into this greenish yellow. And that's evidence that your macrophages in your connective tissues are recycling the organic molecule part of your heme right there in your tissues. It doesn't have to be in the liver and the spleen and the bone marrow. It can happen right there in your tissues. And so that's the progression of a bruise. And eventually your tissues will be all healed. The blood's been all recycled. Your blood vessel got fixed a long time ago. So you can witness this color change right there in your skin. So next time you get a bruise, you can tell your family and friends all about what's happening when it changes from purple to green to yellow. Pretty cool. All right. So now we're going to take a look at red blood cell production. What happens when blood cells are produced in the bone marrow? So we make about 3 million red blood cells per second. Oh my gosh. But what that tells you, that last slide, the activity that we're just doing, that means we recycle about 3 million red blood cells per second. So we are at a balance. However much you take out, because they're worn out, they're done with their 120 days, you have to put in to maintain a healthy blood volume and a homeostasis of blood production and blood recycling. So imagine 3 million, 3 million, 3 million, 3 million being made and being recycled, made and recycled, made and recycled, made and recycled. So our bone marrow is really busy. We've got a lot of stuff going on. Our protein synthesis, mRNAs, ribosomes, tRNAs, all of that, they're just constantly cruising in our bone marrow cells to make red blood cells. And each red blood cell, before it becomes mature, is just going to be pounding out those hemoglobin molecules to what it was 280 million hemoglobin molecules per red blood cell. That's a busy nucleus. Okay. So let me get to the right place in my notes here, red blood cell production. This process is called erythropoiesis. I found a couple pictures from a different couple sources. Erythropoiesis, erythro meaning red blood cell, and poi is like to grow, like a osteoblast was a maker. So poiesis is like a process of making. So erythropoiesis is making red blood cells. It takes place in the bone marrow, the red marrow that is found typically at the proximal ends of your femur and humerus and a couple locations of your flat bones, the spongy bone and your flat bone. Okay, so we'll go through this about a week-long process. We have, starting here, kind of day one, we have what's called a proerythroblast. So it's like the red blood cell stem cell, right? You can see it's clearly got a nucleus. It's a eukaryotic cell. Okay, fits that definition. Then it turns into kind of a few more blasts. So we have erythroblasts, a basophilic erythroblast, a polychromatophilic erythroblast, and a normal blast. So we've got some precursor erythroblasts. We have 3 stages of erythroblasts. And then lastly, we have what's called a reticulocyte, which is the newly formed cell after the nucleus has been ejected. So the normal blast, all this time that nucleus is busy pumping out the hemoglobin, packing that cell full of hemoglobin. And in that conversion between day about 4, 5, 6, and 7, the nucleus is ejected. We have what's called a reticulocyte. It enters the circulation kind of like a loose, sacky, spherical thing. And then it will mature. So that's kind of what this says here, but it's kind of cut off by my face. Mature blood cell that will then be able to circulate for about 4 months, for about 120 days. But again, this is continually happening in the bone marrow. Another thing I wanted to mention in this process, what regulates this process? Well, we can recall a little bit back to the end of chapter 18 when we saw this hormone from the kidneys called erythropoietin. That's where that weird name comes from, because it's the hormone that triggers erythropoiesis. So erythropoietin, when the kidney recognizes low blood volume or reduced oxygen carrying capability, it releases EPO, is the abbreviation for this hormone. You should know that because we just tested over chapter 18. So EPO comes from the kidneys in response to low blood volumes or low oxygen carrying capability. EPO targets your bone marrow, so EPO triggers this whole process to increase the rate of red blood cell production. And so we negative feedback, we have more red blood cells increasing the volume, more red blood cells increasing oxygen carrying capabilities, and we're back to homeostasis. Now, a little bit of application note here, and as we wrap up the discussion of red blood cells. There is a illegal practice called blood doping. You may or may not have heard of that before, but blood doping is where people purposefully inject themselves with EPO illegally to increase their red blood cell production, usually just before an athletic event. So it is an illegal, what's the word I'm looking for? Advantage, right? So it's going to give that person an advantage because they have more red blood cells, they can carry more oxygen, they can let their muscles work faster, which is an illegal, there's a word, I can't think of it. Usually there's a classroom that can give me feedback, but you guys, you're all in your own houses right now. So that's why it's like steroids are illegal advantage in sports. So EPO, blood doping, also is illegal. Now there is a way to do this naturally that is acceptable in sporting circles, is high altitude training. So if you go up to high altitudes, like that's probably why there's a lot of training in Denver and other mountainous areas, is because the higher altitude you go, there's less oxygen in the atmosphere. So as you go there, your body's like, oh, there's less oxygen in my normal breathing. So what am I going to do? I'm going to naturally release EPO, send that to my bone marrow, produce more red blood cells to become adapted to the environment at the high altitude. So if somebody does high altitude training prior to their competition and then come down to sea level, they will have more red blood cells, be able to use the oxygen more efficiently, but it's legal because it takes quite a bit of time. If you inject EPO, you're going to get that right away, but the adaptation that takes place at high altitude training is over a week's long time. And then when you go back down to sea level, eventually about a week or so or two, it'll fade away because your body adapts to the higher oxygen concentrations at the lower altitude. So high altitude training, yes, that's okay to increase your hematocrit, increase your percentage of red blood cells. Blood doping by taking EPO is illegal and is dangerous because in high altitude training, the rest of your body is adapting and it's a slow change. If your heart tries to start pumping extra thick blood around your body because you've injected EPO, then it's going to put a strain on your heart and blood vessels, which could cause some serious damage. Something else came to mind. Oh yeah, so sometimes blood doping is also where you take your own blood out, you get rid of the plasma, and then you just dump the red blood cells in. It's another way to increase your hematocrit, but that also is illegal, I'm pretty sure. So injecting EPO and re-injecting your harvested red blood cells just prior to a sporting event is also unacceptable, I'm pretty sure. I'm not a sports attorney, but I'm pretty sure those things are not good. They're not good for your health overall either. Okay, so that wraps up our discussion of red blood cells. The next video is going to continue our discussion of red blood cells, but focus on blood typing. It's enough of a complex topic that it's going to get its own video. I'll see you next time. Bye.